Alright, hello everyone, thank you so much for tuning in. I'm excited to talk to you about this paper that came out of my PhD. More allogrooming is followed by higher physiological stress in wild female baboons, which was published in Biology Letters earlier this summer. You can access the paper using the link below the video, or if you're feeling lazy, you can just scan this QR code and it'll take you straight to the paper. The work is open access, courtesy of Swansea University, so thank you very much to them. And of course, a big thank you to Biology Letters for inviting me to contribute to this ecology and evolution seminar series. Alright, so let's dive right in with this review paper on the social determinants of health and survival in mammals by Noah Snyder-Mackler and colleagues, where they gathered all the evidence across several mammalian taxa that individuals that are more socially integrated experience fitness benefits such as longer lifespans and higher reproductive outputs. And as you can see circled here in red, the sample size for humans takes the lead by about 300,000, and this is because we have actually been interested in this question for a really long time. So some studies date back to the 70s where they started linking low social integration with increased risk of mortality. And then more recently, studies on non-human mammals have been added to the map. Some famous examples come from long-term baboon studies, such as the one in the Amboseli in Kenya and Moremi, Botswana, where they showed that females that have strong, consistent bonds tend to live longer lives and have more offspring. And so the question here becomes why? What is good about being socially integrated? There are several non-mutually exclusive mechanisms that have been put forward. For example, having social bonds may increase tolerance around food patches, which means that you get better access to resources. Having social bonds might mean that you have better protection against predators because your social bonds will come to your aid when you're in danger. Or social bonds might have thermal benefits because being more socially integrated means you can maybe occupy more central spots in a huddle and stay warmer. And then there is another mechanism, social buffering. And this is the idea that having social bonds can decrease your physiological stress. And this effect has been shown across several taxa, not just mammals. So to explain why social buffering might be beneficial, I have to do a brief detour and talk about the stress response. So the endocrine stress response is highly conserved across vertebrates. And what happens is that when there is a stressor, which can be anything from bad weather to having an aggressive interaction with someone, the hypothalamic pituitary adrenal axis, or HPA-axis for short, is activated. And this releases a range of hormones, including these glucocorticoids, which I'll refer to as GCs throughout the talk. And this is a beneficial adaptive response because GCs help mobilize the energy to cope with the stressor at hand, and it brings the organism back to homeostasis. So this is when the internal state is balanced in terms of blood glucose levels and body temperature and so forth. So the downside, potentially, is that if the HPA-axis is chronically activated, this exposure to GCs can become detrimental to health and can lead to things like immune suppression or reduced cognitive function. Now, it is important to make a little plug here to say that GCs are often seen as the bad guy, that high levels of GCs, or stress hormones as they're sometimes referred to, are bad for fitness. And this is certainly not always the case, and in fact it's usually not the case. And you can read some excellent reviews and meta-analyses about this topic in these references cited below. Nevertheless, in some animals, including in us and other primates, prolonged elevated exposure to GCs has been linked to negative health outcomes. And this may be due to something Robert Sapolsky calls the primate predicament, which is the term he coined to describe the situation that primates contend with daily, namely that psychosocial stressors arise from the social context in which they live, where primates face uncertainty and worry about threats in the environment, even if they're not actually happening to them in that very moment. So this might activate the HPA-axis in a maladaptive way, leading to exposure to GCs over time that can become detrimental to health. And so in this context, it is interesting to think about social buffering as a sort of counterforce that can shield the organism. Now, social buffering has been documented to occur through the mere presence of social bonds, but social touch does seem to play an important role as well. We know this intuitively, having gone through the pandemic. Many people reported suffering from this lack of physical contact, and scientific studies back this up. For instance, the study by Ditson and colleagues found that HPA-axis activity in women about to be exposed to a stressful situation was only attenuated when having support through physical contact with their partner, and that this effect was not found if the partner was simply providing support verbally over the phone, for example. Now, what's interesting is that this effect need not be limited to only partners or friends. It can also occur in response to social touch from a stranger. For example, when you receive a massage, this has been linked to lowered GCs as well. So the mechanism behind this is likely linked to oxytocin and other endorphins that are released in response to social touch, which then downregulate HPA-axis activity. And so this could be one potential way in which social bonds provide health benefits. And if we look at social touch in primates, which we refer to as allogrooming or simply grooming, we also find a suite of evidence that links grooming to lower GCs. This is not necessarily surprising, especially for the recipient of the grooming, where we typically assume that it's relaxing to be groomed. But interestingly, it is also found that giving grooming can be related to lower GCs. So the tricky thing with these studies is that they link broader patterns of grooming with average GC profile. So it's not clear whether there really is a causal link between the social touch and the reduced GCs. There is some evidence coming from the captive and semi-free-ranging primate work. Here they showed that recipients that are being groomed have a reduced heart rate in rhesus and pigtailed macaques, and in Japanese macaques they showed that both the recipient and the donor of the grooming had lower behavioral indicators of stress, so less self-scratching and less yawning after grooming. So can we study these short-term effects of grooming in the wild? For good reason. Research in captivity tends to focus on proximate mechanisms. In the lab, many factors can be kept stable, so you can eliminate certain sources of noise like nutrition or climatic stress, which would affect the GCs. Also, the social environment can to some extent be engineered and manipulated. In contrast, studies in the wild often take a more zoomed-out approach and address ultimate questions, like, does having higher rates of positive social contact correlate with higher survival? But zooming in and studying the proximate mechanisms in the wild is important because even if we do find support for grooming reducing GCs in captivity, for example, we won't know if this relationship holds in the wild. So, to my knowledge, there is only one study that has actually studied this mechanism in the wild and looked at how grooming affects GCs directly, and that's this study by Whittock and colleagues on chimpanzees. So what they did here is they followed individual chimps for 6 hours in both stressful and neutral contexts and recorded every grooming interaction, as well as collected as many urine samples as possible. And what they found was that grooming with a close bond partner reduced urinary GCs relative to when they did not groom with a close bond partner. And so this is really amazing work, but it's also challenging. Working with non-invasive samples like urine or feces, which are of course opportunistically collected, means that you don't always have the data to directly match the grooming to the subsequent GCs unless you have a very focused protocol like the one that they had in this study. So during my PhD, we explored a new method, namely tracking colors that can record behavioral data continuously through accelerometers to study grooming hormone links in the wild. I will tell you a little bit more about accelerometers in a minute, but before that, I just want to lay out the benefits in this table. So imagine if we had technology that could continuously record grooming data from a collar. It means that opportunistic samples could be matched retrospectively to the grooming that happened beforehand, or indeed you could travel forward in time and match the grooming that happened after the physiological state. And this is in contrast to traditional methods where you would typically have a rate of grooming that would be correlated to a sample collected around the same time. On top of that, if you have continuous data, you can extract things like total amount of grooming, rather than having rates of grooming which are typically based on a focal follow of maybe 10 to 30 minutes per day, like the one that I'm collecting here on the baboons. And so we tested this new method, as you already know from the title, in chacma baboons. So baboons offer an excellent study system for examining the physiological consequences of grooming. They devote more time to grooming than any other genus of primates, and they have been proven to be a tractable system for the exchange of grooming for commodities such as reduced aggression and tolerance, infant handling, alliance support, and trading it for grooming itself. We also know that grooming and grooming-related network metrics have been linked to changes in GCs, and importantly, for the motivation of the study, that cumulative lifelong exposure to GCs is linked to reduced lifespans in female baboons. And the particular chacma baboons that we studied live on the Cape Peninsula in South Africa. The troop was called the Dagama troop, and this was named after the urban area in which they would often sleep. The troop consisted of approximately 50 baboons, with 19 adult females and 2 adult males, and we studied them between July and November 2018. So let's talk about these collars. They were built at Swansea University and contained both a GPS and an accelerometer. The accelerometer is this little device here in the second picture, which was developed by Wildbytes, and it records triaxial acceleration data. So, as the name suggests, it records acceleration in 3 directions: heave, up and down, surge, front to back, and sway, side to side. And you can think of these signals as creating a signature of the behavior that the animal is doing. And if we have an accelerometer on an animal, we can sort of continuously record what that animal is doing. And so you might be wondering, what does grooming look like in acceleration data? And I will show you in this video. So here is a video of a female grooming another female. The collared female is the one up on the left. That's the one that we're seeing the data from. And here she is giving grooming, and you see a little bit of wiggle around these 3 acceleration signals. She then stops, turns around, and becomes the recipient of the grooming. And so here we see both the change in posture reflected in the ACC signal, and we also see that the movement around all 3 axes becomes reduced as she becomes the recipient of the grooming interaction. So by collecting many of these videos and labeling them back in Swansea, we could create this data frame, which was about 23 hours of video data, where for each second we have the behavior the baboon was engaged in and the associated acceleration signal. And with this data, we could feed it into a supervised machine learning model, here a random forest model. And what this model does is essentially learn through many iterations what signals are associated with what behavior, which means that once the model is trained, you can apply it to new, previously unseen data and classify the behaviors that are happening second by second, including grooming. So, we are getting closer to the meat of the talk, the main question: does grooming directly reduce GCs in a wild system? We now have the grooming data from the collars, but we still need the GCs. So to measure GCs, we collected hundreds of fecal samples from our baboons. The samples were freeze-dried at UCT and then exported for fecal glucocorticoid metabolite analyses at Swansea University and the German Primate Center. And so for more details on the methods, you can read this paper here. For the purpose of the analysis, we matched 185 of these fecal samples from 10 different females to the associated collar data. So how did we do this? The first thing to establish was the time lag. So when you collect a fecal sample and measure the GCs therein, it represents the GCs that were circulating from a period in the past. And there's an excretion time lag that will depend on species, body size, diet, metabolism, and so forth. And so if we look at the literature, we know from a closely related yellow baboons that the excretion between labeled steroid hormone infusion and the excretion in the feces is about 36 to 48 hours. And this matches closely with what we found in our biological validation, where when we deployed the collars 2 days later, we saw an increase in fecal GCs. So with that in mind, we use a 2-day time lag to travel back and capture the relevant behavioral data. And now the second question, which in my mind is a little harder, is how big should this grooming window be? So we wanted to strike a balance between including enough time to capture the grooming that took place, because unlike an acute stressor, which can be something that happens in a matter of seconds, we assume that grooming is a cumulative experience. And on the other hand, we didn't want to include so much data that the grooming that happened was no longer relevant for the hormonal state that we were capturing in the sample. And so what we did is follow the example set by 2 previous papers that used this hormone matching approach. One was the chimpanzee paper that I mentioned earlier, and essentially they matched the time lag period duration to the duration that they sampled. So here we consider grooming over a 2-day window. Right, so we're finally at the question of the paper: is grooming followed by lower fecal GCs? So on this figure, you will see on the x-axis the total amount of grooming that happened in this 2-day window in hours, and then on the y-axis you'll see the fecal GCs expressed in nanogram per gram. And in blue you will see the receiving grooming, and in orange the giving grooming. And so we found the opposite. We found that rather than grooming being followed by lower fecal GCs, it was followed by higher fecal GCs, and this was true for both giving and receiving grooming. So this left us a little bit perplexed, and we wondered if it was possible that what we were capturing was not the effect of grooming, but rather the effect of some underlying stressor, which was causing both the increase in grooming and the increase in fecal GCs that we were measuring. And this would be in line with the tend and befriend phenomenon, which is particularly prevalent in female primates and describes the behavioral response of seeking out affiliative interactions under stressful conditions. This has been shown in female baboons as well, who increased their grooming rates and their grooming network after losing a close bond partner to a predation. So we flipped the analysis and asked, does fGCs predict the time spent grooming in the lag window, assuming that if this were a period of high physiological stress, that it should also trigger more grooming in the lag window? But we found no support for this. So giving grooming and receiving grooming in the lag window were both unaffected by the GCs in the sample. So if we had to summarize this, we find evidence that grooming is followed by higher fecal GCs, but not that higher GCs are followed by more grooming, which leaves the question, is grooming in and of itself stressful? And there is some evidence to suggest this might be the case. In Barbary macaques, grooming was followed by more self-scratching rather than less, which was contrary to the predictions that Sample and colleagues made. And if you think about the act of giving grooming in a hierarchical system, such as the one these macaques, the baboons, and many other primates live in, you're essentially at risk of aggression or displacement constantly, especially as a more subordinate individual. And in a paper by Garcia and colleagues on vervet monkeys, they described the tolerance incurred from grooming as the sand in an hourglass, where the amount of grooming you gave was proportional to the amount of time the more dominant individual would tolerate you before returning to beating you up again and stealing your food. So if you put it this way, it makes sense that the act of grooming as an appeasement strategy may not be relaxing in and of itself. Also, being engaged in grooming opens you up to aggression, both from other individuals or from the recipient itself, who may be frustrated at the end of the grooming bout. What was more surprising here was that receiving grooming was also associated with increased GCs. So typically we think of being groomed as relaxing, especially if we view it through this slightly anthropomorphic lens of receiving a massage. But if we think about it, the recipient may also be exposed to the same risks of aggression and interference from bystanders. Moreover, in our sample of 10 females, 7 of them had infants. And it's widely documented that female baboons are very attracted to the infants of other females and will try to handle them. This was similar to what we saw in our troop, where the females would display this almost feverish compulsion to hold infants from other females. And this also means that the mother is being groomed by females not necessarily of her choosing, who are just trying to get a hold of her baby. And this unsolicited attention has previously been linked to increased GCs in baboons. So let's zoom out from the primate world and consider some other interesting parallels in other systems. So here in these cleaner fish, the authors show that cleaner fish who tend to both predatory and non-predatory client fish had an increase in GCs before cleaning a predatory client. Nevertheless, they would still do so, and they would approach these clients quickly and clean them for longer. And the authors hypothesized that one of the benefits of this was that they reduced their own chances of being predated. Finally, our findings are also in line with the social bond testing hypothesis, which posits that closely bonded individuals should sometimes engage in risky interactions, such as presenting genitalia to one another, as these two spotted hyenas are doing in this photo, and that this strengthens the bond, because by engaging in a short-term risk, it signals a commitment to the long-term bond. So these examples highlight the contrast between the short-term cost of a behavior and its long-term benefit. And if we place this in the context of our findings and we revisit our troop for a second, we can take a look at this final result, which we put into our supplementary material. And this is the long-term correlation between the total grooming rate over about a month and a half and the average fecal GCs for 7 females. Unfortunately, we had to exclude 3 of our females because they were pregnant, and this increases fecal GCs and cannot be controlled for in a correlation. However, we find a negative relationship, which is not statistically significant, but it is in line with the sort of expected direction that we were expecting to see, and also with other papers that link grooming and GCs in this more zoomed out way. And so further studies will of course be needed to see if this result can be replicated in other study systems and across different contexts, as here we are just investigating this relationship with 10 females over a short time window. But if I had to leave you with a take-home message, it would be this one. Is grooming a short-term cost for a long-term benefit? One in which the grooming females engage in a potentially risky activity where they are exposed to potential aggression, both from the baboon they're grooming or from a bystander, but in the end, they benefit from these grooming exchanges to gain access to benefits such as tolerance or protection in a fight, which then in the long run results in this lowered average GC that we see. And with that, I just want to take a moment to acknowledge that I could have never done all this work by myself. I was supported and supervised by Ines Vuurtbouwer, my primary PhD supervisor, as well as by Andrew King and Justin O'Rian. I had the pleasure of conducting the fieldwork with Anna Bracken, who was another PhD student on the project. We are also very grateful that Michael Heisterman was on board as a co-author for this paper and did the fecal GC analyses. I have a lot of people I'm grateful to back in Swansea who helped build the collars as well as interpret the accelerometer data. We had help in the field deploying the collars. We had funding both from ASAB and from Swansea University in the form of PhD scholarship. And we are finally also very grateful to 3 field assistants, Charlotte, Lucy, and Fran, who collected a lot of baboon fecal samples. And with that, I want to say thank you to you for listening, and if you have any questions, do get in touch. My email, my Twitter, or X, I guess, and my blue